Sorry, just can you see my screen, Francesca?
We see the first slide of your presentation with the
Ah, okay,
title.
perfect. Okay. Okay, so once more, the title of my talk is Biomolecular Force Fields, Recent Advances in Non-Standard Amino Acids and Nucleic Acid Development. This is the outline. Today I will speak about biomolecular force fields, especially for its historical evolution. I will speak about challenges in parameterizing non-standard amino acids. And I will speak in details about methodologies for the developing force fields for the TOAC, amino isobutyric acid, and aminocyclohexane acetic acid. Then, since we hear a lot about nucleic acids force field simulations, I will assume some challenges in nucleic acid force field development. I will speak about some advancements in polarizable force fields, and I will mention some future directions in nucleic acid force fields. Biomolecular force fields are computational models that are used in molecular dynamics and related simulations to describe the potential energy of a system of atoms. They simulate the physical forces and interactions such as bond stretching, angle bending, torsional rotation, mass interactions, and electrostatic forces that govern the structure dynamics and energetics of biomolecules like proteins, nucleic acids, and lipids. In biomolecular simulations, force fields are essential for studying protein folding and dynamics, DNA, RNA structure and interactions, ligand binding and docking, and for example, membrane, they're important for the membrane protein simulations. Some early models of the force fields are focused on the reproducing basic structural features and empirical data. Initially, they are limited to small peptides and globular proteins. Some historical development of force field is AMBER, force field 94, CHARM 22, and OPLS-A. Next years, several generations of force fields have been developed for nucleic acids with continuous improvements aimed at enhancing accuracy, especially for RNA and DNA dynamic behavior. These key force fields include the CHARM 27 and CHARM 36. Next generation of amber force field represents a significant improvement of the backbone side change, providing a better accuracy in reproducing key protein structural features, particularly a better balance of the secondary structure alpha helix and the tryptophan structures. And the famous force field is force field 99SP, which contains the first parameters which are responsible for the better balance of the secondary structures. Key force fields include, besides force field 99 for the nucleic acids, is PARM BSC1, OL15 for RNA, which contains some parameters which are responsible for better description of sugars, and for the DNA, it's OL24, the recent refinement. Modern force fields, or let's say force fields from the 2010s and to nowadays, are focused on accuracy and transferability, the incorporation of polarizable force fields, some good models, extensive validation with a lot of experimental data and quantum data available, and of course use of machine learning for parameter optimization if possible. Uh, the main and perhaps the most important input of molecular simulations of biomolecules is force field. Developing force fields for biomolecules is crucial aspect of computational chemistry and life sciences, and these force fields are used in molecular simulations to predict the behavior, structure, and properties of biomolecules under various conditions. A force field is a collection of equations and associated constants, which are designed to reproduce molecular geometry and selected properties of molecules. It is based on Born-Oppenheimer approximation, and where in the force field method, we renounce to an explicit treatment of electrons, and we calculate the energy of the system as a function of the nuclear positions only. Now I will speak about challenges in the modeling of the non-standard amino acids with the amber force field, amber-based force field. Modeling non-standard amino acids in molecular simulations presents several technical and methodological challenges. And these amino acids are not part of the 20 standard amino acids encoded by the standard genetic code, and they often include some post-translational modifications, some synthetic analogs, or contain some chemically modified residues. Standard force fields like amber, charm, or OPLS are designed for the 20 natural amino acids. So non-standard amino acids often are missing from this parameter data set. And since they have some unique size change or modifications, that's the reason they are not covered with the standard force fields. Further, there is also incompatibility with some automated many tools which are implemented in the NAMD, in GROMACS, in AMBER, like LEAP, which fail when you want to model and to find parameters for the non-standard amino acid residues. This requires some manual derivation of bonded and non-bonded parameters, such as charges, diedrals, or Lennard-Jones parameters. Now I'm going to talk about force field refinement of amber parameters for the description of alpha-alpha dealkylated peptides. Here are shown non-standard amino acids for which the parameterization has been applied. The simplest and most extensively investigated member of this class is amino isobutyric acid, which has been shown to effectively stabilize conformations in the region of tritone helices and alpha helices. Amino isobutyric acid is structurally similar to alanine with a methyl group instead of hydrogen at the place of alpha carbon. TOAC is a member of the family of C-alpha-alpha desubstituted glycines, characterized by a 6-member heterocyclic saturated side chain containing paramagnetic nitroxide radical, which is presented here in the graph. And the final aminocyclohexane acetic acid also belongs to the same family. The incorporation of C-alpha tetrasubstituted alpha amino acids into peptides represents an effective and versatile strategy to restrict the conformational freedom and to stabilize defined secondary structures, alpha helices or tritone structures. As it is well known, the behavior of these rigid amino acids differs substantially from standard monosubstituted amino acids, and their description at the force field level is not obvious. Further complications may appear when nitroxide probes are inserted into peptide chains, like in the case of the TOAC. We were interested here in providing reliable and an accurate force field at amber level of theory, able to explain the role of the steric hindrance, around the C alpha carbon atoms, and this steric hindrance governing helical structure and flexibility of the C alpha tetrasubstituted peptides. The nitroxide spin labeling method is widely adopted in the field of protein chemistry. In almost all the studies, proteins were labeled by coupling nitroxide-containing molecules to reactive groups of amino acid side chains, mostly to the SH group of cysteine. Once nitroxide spin labels have been attached to a protein or enzyme, a wealth of information can be collected. If a single probe is presented, this essentially concerns the rotational diffusion and wobbling of the nitroxide. Dipole-dipole interactions with proton nuclear spins and possibly the quenching of excited states of chromophores. If more than one probe has been introduced into the proteins, properties like dipole-dipole interactions and static and dynamic spin exchange between nitroxide also can be determined. Nitroxides are extremely interesting from the chemical point of view, since in contrast with the other free radicals that are highly unstable, the NO group is stable under a variety of physical chemical conditions. This allows to obtaining of their different spectra in different solvents, including water, in a range of pH and temperature. Multi-frequency electron paramagnetic resonance combined with side-directed spin labeling is a powerful spectroscopic tool to characterize protein dynamics. A multi-frequency EPM distinguishes between different motions of spin label proteins. Amino acid talk was the first spin label probe incorporated peptides by means of a peptide bond. The incorporation of C alpha substituted alpha amino acids into peptides represents a good strategy to restrict the conformational freedom and to stabilize defined secondary structures. Many of studies were performed making use of EPR spectroscopy, but other physical techniques such as X-ray, crystallography, and MR also have been employed. Now I will mention the recent development and extension of amber force field with the parameters for the accurate description of large and flexible nitroxide-free radicals in condensed phases. The parameterization was based on a training set of 7 medium-sized nitroxide radicals, which are presented here and which are representative of the different ranges of pyramidalization for NO moiety. New atom types related to this nitroxide radical were added to standard amber force field 99 SB as one which was the best in that time. And in dash line, explicit lone pairs were placed on the nitroxide oxygen. Here are in dash line placed lone pairs, and this represent explicitly introduction of the lone pairs allowed to better mimic the interaction between NO moiety and protein solvents molecules. The refined parameters were further applied in an integrated approach where the magnetic properties are determined by using parameters which were derived for the nitrogen, and they were calculated at level of theory and also at QM level of theory. In these tables, you may see that a very good agreement was observed in different solvents, in vacuum, toluene, and water. This is now general form of the amber force field that can be used for so the first term is related to the deviation from the bonds, where chi r eq is are constant. Then the second term is related to the deviation of the angles, the third related to the deviation of the dihedrals, and the last two are related to the explanation of the non-bonding interactions between atoms in protein molecules. So all the empirical parameters are labeled in yellow. So now I will first speak about the force field which are necessary to determine the deviations of the bonds from their natural values. So many force fields use just harmonic potential, and in some cases some higher order terms can be present, but in number mostly this in number, this harmonic potential with one term is enough, where Kb is force constant, B0 is reference bond length. In such case, in some cases, Morse potential, which is more accurate, also can be used, but with a higher computational cost. The angle band term refers to the bond angle deformation of energy in molecular simulations, particularly in force field models used to describe flexible molecules like amino acids. And usually a quadric polynomial term also is sufficient with the parameter k theta and theta 0, which is ideal angle, which is without any string. Angle bending affects angle bending modeling angle valence angle bending terms affects secondary structures like alpha helices and beta sheets and the overall folding of proteins. Even small angle distortions can disrupt hydrogen bonding or steric interactions, changing the structure or function of a protein. Torsional terms are models that can be usually described with the periodic and the cosine function, which is presented here. In this equation, Vn is force constant, n is periodicity of the angle, and determines how many peaks and wells in the potential are. Phi is torsional angle. Gamma is phase of the angle. Usually it is 0 and 180. Degrees. Determining the torsional term, also called the other term, for amino acids is a crucial step in building or refining a force field for molecular modeling. And this term captures the rotation around bonds, especially in the backbone and side chains. Which is essential for modeling conformational flexibility of proteins. For modeling conformational flexibility of proteins, it is necessary to involve multiple torsional terms, and usually up to 3 is enough. For example, while we develop force field, refined amber force field for non-standard amino acids, we also involve this multiple torsional terms in order to achieve good conformational flexibility of the amino acids with cyclohexane rings. usually, if you would like to determine parameters for single bond, you need to assign 9 torsion, and for the double bond, you need to assign 4 torsions. To determine torsional parameters for a molecule, so you typically use quantum mechanical data, and here is some practical overview of the process that you have to perform. So the first step is to identify the torsion, choose the dieder torsion you want to parameterize. In amino acids, torsions include the backbone Phi and C. Then you perform a QM torsional scan. You freeze the dieder angles from 0 to 360 degrees in 15 degrees increments. Then you optimize all other atoms at each step using DFT. After that, you record the total energy at each point. The third step is to fit the torsional potential to the QM data, to adjust the parameters VNN and gamma that I mentioned in the previous slide, and to minimize the error between QM and fitted energy. And at the end, you have to compare your result with experimental available structures with some Ramachandran plots, or if necessary, you have to adjust your parameters and then to repeat the step 3 and 4. In many cases, so you need to involve also the term which is called angle plane term, and the angle, this term enforce polarity around to sp2 carbon atom, and this out of plane bending energy is can be described with also with harmonic function with the parameters KOO, force field constant, and alpha is here improper torsional angle. Deviation, it means deviation from the planar angle. Alpha 0 is equilibrium angle, typically 0 radian when the system is perfectly planar. For more accurate representation of the potential energy surface, for example, for vibrational frequencies, this requires interaction, many terms, interaction between terms, between stretch and bend, bend and torsion, etc. Now I came to the determination of the non-bonding parameters. So the non-bonding term is consisted of the terms which is which reflects the van der Waals interaction and electrostatic interactions. Hydrogen bonds are usually implicitly introduced in these two terms, and it is not easy to manage hydrogen bond implicitly, but just explicitly, and you always have to have enough experimental data to validate the hydrogen bonds that you observe in your molecular dynamic simulations with the real situation in the system. And this is the Lennard-Jones, the most widely used potential, 12.6, and this is the graph which followed this equation. In this equation, epsilon is well, sigma is the distance which corresponds to the minima, and r is the distance between the atoms. The first part is related to the repulsion, and the second part is related to the attraction, which are long distance interactions. Sometimes for the defining non-bonding interaction, Buckingham potential, which is more accurate and more realistic with exponential function, can be used. but since it is more expensive, it is not widely accepted in the regular force fields like amber, charm, and OPLS. Electrostatic interactions are fundamental in modeling proteins since they consist entirely of charged species like nucleic acids and its behavior is largely governed by Coulombic forces. In molecular simulations, these interactions are calculated using Coulomb law, with placing partial charges on each atom with the distance between them label R, and with the dielectric permittivity label epsilon 0. So this is the roadmap of the force field parameterization, which we perform for the refined amber force field for this non-standard amino acids in order to model the peptides containing these non-standard amino acids. So the idea was to introduce in standard amber, in current amber, as less as possible parameters in order to achieve good agreement between QM data and available experimental data. As a model systems, we use the dipeptides of the aminocyclohexane acid, TOAC, and the aminoisobutyric acid as the small systems with with the good representation of the whole molecules in which we were interested. After atom time assignments and determination of partial charges, you have to determine all other parameters necessary for bonds, for modeling bonds, angles, torsions, improper or van der Waals interactions. And then to optimize these dipeptides at QM level of theory, we use for this PB0 DFT calculations at PB0 level of theory, or literally with the inclusion of the dispersion corrections. Then we compare our results our results developed we can calculate at level of theory energies and geometries for these dipeptides with QM data, and until satisfactory agreement between QM data and data, we do this refinement of the force fields. And if we are satisfied with the results, we go further to the force field validation by using, by performing MD simulations and by comparing our results with as many as possible experimental data, X-ray, IR, EPR spectra. In this slide are presented the new atom types that were introduced in the standard amber force field, which are around the C-alpha tetra substituted carbon atoms. So we basically introduce two new types for the carbon atom and for the hydrogen, which was very near to this C-alpha carbon atom. Also in dashed line, explicit lone pairs are placed on the nitroxide oxygen. So this high specificity of atoms was necessary because it allows us to introduce diedral angles and valence angles, which were responsible in governing helical structures of the peptides that we wanted to simulate. This is the slide related to the initial structure that we generated for the force field development. Since we had the cyclohexane ring, we for the force field development, we took in account all possible conformations of cyclohexane rings, all possible chair and twisted conformations. And while both conformations were not considered, and by taking into account of relative positions, equatorial or axial, of the substituted carboxyl group, we came to the 27 structures as a starting points for the TOAC dipeptide, for example. Partial charges were obtained by using a RES program, which is implemented in Amber, and charges were fitted against the QM calculated electrostatic potential. Here, the chemically equivalent atoms were set to have the same charges, while the total charges of the acetyl and N-methyl amino groups were constrained to be zero in order to allow incorporation of the data set parameters in current Amber force field. We also had to develop some van der Waals parameters, which are related to the atoms around this C alpha carbon atoms, since these van der Waals parameters significantly affect the distribution of the secondary structures in different solvents. So we had to introduce parameters, especially this effect of necessity of introducing new van der Waals parameters was seen in the peptides with the higher number of amino acid residues. So we optimize van der Waals parameters by starting from the standard Amber parameters, and we just refine them based on the high-level quantum chemical data and available experimental data. So the parameters were fitted, these bonded parameters were fitted by using the JOICE program. The main goal was to find, with respect of the reference QM computed data, the best parameters by which can minimize the following function. In this equation appears some Hessian matrix that we had to calculate at QM and the force field level of theory. And they are evaluated at the absolute minimum. And this is in this graph a result of our force field refinement. We had a lot of QM data for amino isobutyric containing peptides with the residues from 1 to 33. And here we do comparative QM and MM calculations of the relative stability between alpha helix and tritone helix. We observed the root mean square, energy root mean square deviation of all peptides with the new force field assumed 0.37, whereas with old Amber, this root mean square deviation of the relative stability of the tritone helix to alpha helix in comparison with QM data was 7.45. So this means that the new force field significantly improves the quality of the parameters for amino isobutyric acid. We also took into account solvent effects and performed the same simulations in PCM and chloroform. And as a result, we observed that solvent effects lead to a preference for the tritone helix, which is the most favorable and low polarity solvents. Whereas in water, for example, alpha helix is more stable due to the different hydrogen bond network, which is more stabilized by water in alpha helix. Comparison in this table as a standard, we perform comparison between energies and geometries of the selected structural feature of the aminocyclohexane acetic acid, and the same study we did for the TOAC dipeptides. We observed starting from 27 initial conformers, we observed 15 stable conformers at DFT level of theory, and also at level of theory. And here is the results of the comparison of their energies and geometries. Also an energy root mean square deviation over all conformers were 1.28 with the new refined force field, and with the old force field amber was about 4. Also, there is some significant improvement, especially for the proteins that contain beta residues with the phi and psi angles 180 degrees. Also, we observe very good agreement in this selected phi and psi values in comparison with the QM data, where the differences are less for the torsional angles, less than 10 degrees, and for the valence angles less than 3.5 degrees. This is the table, the similar table only for the TOAC dipeptide analogs. Also, we observe 15 different stable conformers with an energy root mean square deviation very low, about 1.32. And this graph, you may see superposition of the selected TOAC dipeptides together with the dipeptides which were optimized at QM level of theory, and a very good agreement was observed with the average root mean square deviation for 15 conformers about 0.04 angstroms in geometries. Further, we also perform a lot of molecular dynamic simulations as we were satisfied with this first step of comparing our results with quantum mechanical data. Then we were interested in molecular dynamic simulations of double spin label peptide, which is presented here in the slide. The oligopeptide is labeled with 2 nitroxide radicals in the form of at the position of 3 and 6. And from experimentalists, it is known that it falls in a 310 helical structure in non-aqueous solutions and in alpha helical structure in aqueous solution. In these slides are presented some experimental data where experimentalists measure the distance between 2 nitroxide radicals for alpha helix is 9.2 angstroms, and for 310 helix is 6.8 angstroms. This information was very important for us when performing molecular dynamic simulations, where we also measuring the distance between these 2 nitroxes and were able to detect the secondary spectra. So in this graph are presented results of molecular dynamic simulations in vacuum, where we observed by measuring this distance between 2 oxygen atoms of nitrogen radical to be about 6, in average value was about 635 angstroms, which clearly indicated the presence of the 310 helix in the vacuum. The similar simulations we also perform in water, where we observed that in water, in both of the structures with the helical preference for the alpha helix. And this is also an overview of the further molecular dynamic simulation that we perform by changing the length of the amino isobutyric acids, temperature, and solvents. So we perform simulations in water which reflects one polar solvent, and DMSO, which reflects one non-polar solvent. And as a result, we observed, for example, for one heptapeptide, that in the at the higher temperature and at the DMSO, tritone helix is more stable, whereas at lower temperature and in the more polar solvent, alpha helix is more favored. And this result is in agreement with the experimental data, available experimental data. Now I will shift to some more let's say application of the force, our refined force field for the higher peptides. For this purpose, we use phospholambam 50 protein, which is responsible for the cardiac transport of calcium, and it consists of 52 amino acid residues. It is connected of two parts, one which is cytoplasmatic and the other one transmembrane, which is more rigid. In this graph are presented some experimental results which initiated our work here. Experimentalists actually label these peptides with the TOAC at different positions, at positions 0, 11, and 24 and 46. And then they detect some signals in EPR spectra, and these signals are connected with their structure, and structure is also connected with its function. So on the right side of this graph, you may see that TOAC 46 phospholambam gave only a single spectral component, which is indicative of highly ordered transmembrane helix. Whereas TOAC at position 0, 11, or 24 presented two-component spectral corresponding to two conformational states. Phospholambam cytoplasmatic domain is in dynamic equilibrium between an order conformation, which is in direct contact with the membrane surface, and a dynamically disordered form which can be detached from the membrane and can interact with its target, like Cerca, for example. So here, transmembrane part is more fixed, and the cytoplasmatic part is more flexible. So we wanted to see all this conformation of this phospholambam, and for this purpose, we performed simulation for the native phospholambam without any labels, introduction and any labels of the TOAC amino acids. And we also labeled TOAC at different positions of the phospholambam. And here you may see the result of the molecular dynamic simulations for the native phospholambam and TOAC label phospholambam in position 11, where we observed that TOAC label phospholambam was more flexible, whereas the native is more rigid. And how we measure this flexibility? So we measure actually the angle between three amino acid which is determined by three amino acid residues at three different parts of phospholambam, and we measure this angle versus time, where we observed actually we detect transitions between here on the right the order to disorder conformations, and this disorder conformation actually is responsible to interact for the interaction of the phospholambam cytoplasmatic part with its target, with Cerca. Introduction of the TOAC amino acid into phospholambam sequence has negligible effects on the secondary structure as we observed from Ramachandran plots. So now I will just mention some and summarize some challenges in nucleic acid force field development. As you heard here a lot about the modeling of nucleic acids, I will just summarize that the main challenges are charge density issues. Nucleic acids, they have highly negatively charged bonds due to phosphate groups, and this charge density complicates simulations by causing significant electrostatic repulsion. Then base stacking and hydrogen bonding also can complicate to the force field development, which is especially base stacking some new feature in comparison with the protein. So hydrogen bonding between base pairs, adenine, thymine, guanine, cytosine, and pp stacking interactions between adjacent bases are critical to nucleic acid stability, and capturing these interactions accurately is essential for modeling helical structure and hybridization. Nucleic acids possess very high conformational flexibility. For example, RNA can adopt various secondary and tertiary structures, which which must be accurately captured in simulations, while DNA is more rigid, also exhibits flexibility through bending, twisting, and supercooling, and depending on the environment can adopt different structures beside the well-known double helix. Current force fields also may fail to accurately represent nucleic acid interactions with ions and ligands. Besides these challenges, I will also add that there is also validation difficulties of the force field for nucleic acids, which requires high quality experimental data, NMR, X-ray, and a lot of quantum mechanical data, which are not available since these systems are, in some cases, not available since systems are huge. Then nucleic acids are large, so polarizable models are slower in comparison with the system that are not so large. Extensive solvent and ion effects also lead to higher simulation costs. Then also there is one challenge, transferability of parameters when going from one force field to the other one. Parameters may not be generalized when going from one nucleic acid forms to the other. So this can be also a problem. This is more challenging than in force field development of the transferability of parameters for proteins. So as a solution, polarizable force field, of course, has to be used for the modeling nucleic acids, and they are crucial for accurately modeling biomolecular interactions by taking into account electronic polarization effects, which arise from the distortion of electron clouds in response to nearby electric fields. And they can better explain the dynamics of DNA and RNA, ion binding, and also the base stacking and hydrogen bonds. For the polarizable force fields, three primary approaches are used in developing this type of force fields. First one is fluctuating charge models. In these models, atomic charges are allowed to fluctuate based on the local environment, reflecting the dynamic nature of molecular interactions. And this approach helps capture polarization effects in nucleic acids where charge states can change with local conformations. And induce dipole models. And these models treat atoms as point dipoles, whose moments can be induced by external field, given this with this equation, where alpha denotes the polarizability of the site, and we signify the external electric field acting on it. Induced dipoles also influence the overall electric field, making polarization a non-additive process that is typically resolved iteratively using a self-consistent approach. Induced dipole moment is usually calculated based on the actin field, providing a more accurate representation of intermolecular interaction, especially for systems involving charged groups like those found in nucleic acids. The third method is classical Drude oscillator models. For example, they are implemented in CHARM. And this method introduces additional degrees of freedom by simulating polarizability through virtual particles, which are denoted as Drude particles, and they are bonded to classical atoms. These Drude particles are usually linked with these classical atoms via harmonic bonds, with the force constant being universally related to the atom's polarizability. The Drude model can be used successfully in various simulations, including those of nucleic acids, allowing for better representation of the electronic environment and interactions with, for example, with ions. About coarse-grain model, I will just summarize what is already said in more details by Francesca and Atto. Coarse-grain models are well used for the, as you heard in this morning, in the simulations of the nucleic acids. And it's a simplified approach that reduce system complexity by grouping multiple atoms into larger bands representing key molecular components. The number of atoms represented by a single bed in a coarse-grain model varies from one model to another, and reflecting a balance between computational efficiency and accuracy. As Francesca already said, coarse-grain models can be broadly classified into two types, top-down and bottom-up. Top-down coarse-grain models are parameterized to reproduce microscopic properties, while bottom-up models aim to reproduce microscopic properties and top-down macroscopic properties. And this picture is an example of coarse-grain mapping of the human telomeric G quadruplex that is coarse-grain model, which is developed by Aksonen and Rebic. As of here are presented some future directions in which a nucleic acid force field development has to go. So for the nucleic acid simulations, it is necessary to further improve electrostatic development of polarizable force fields, such as Drude and fluctuating charge model, also has to be improved. And to account for electronic polarization, which is especially important in highly charged systems like nucleic acids are. Then it is also necessary to reparameterize the angles. Focus is on improving backbone and the address for the sugars to better reproduce experimental NMR and quantum mechanical data. And then it is also necessary to refine base stacking and hydrogen bonding. More accurate modeling of base pair stacking interactions, non-canonical base pairs, and loop regions using high-level quantum mechanical data. To overcome all these challenges, machine learning techniques also can be used to enhance nucleic acid force fields by improving all these by improving parameterization and predictive capabilities, which are comparable with experimental data. And of course, scalability improvements will also facilitate the simulation of larger and more complex biological systems. So in conclusion, I will say that force field parameters should be system-specific, derived from accurate quantum mechanical data or experimental data, while adhering to the core principles of additivity and transferability. In most condensed phase simulations, pairwise additivity is applied, where the total potential energy of a system is the sum of intermolecular and intramolecular interactions. Transferability allows force field to predict behavior of different systems, so this goal also has to be satisfied. As a promising approaches are those that use Drude particles and classical Drude oscillator. They offer a computationally efficient solution, especially for systems where polarizability is a key. So with this, I will end this lecture. And at the end, I would like to thank to the organizers of this training school. Again, I would like to thank to Sergi, to Francesca, Pilar, Christina, and thank to COSI, Cost Action, for support. And I would like also to thank you for listening to this lecture.
Thank you very much, Sonia. You have been on time. There are 5 minutes for the question. And I will start with some curiosity.
Francesca? Francesca, can you ask the question?
Oh, okay. Sorry, I think I have the microphone off. Can you hear me now?
Yes, yes, yes.
Thank you very much. I have a curiosity concerning how you sample the conformational space of the 6 atom rings, because you found like 32 minima, but I was wondering how it was performed, the sampling, because if you have a linear chain, you can scan the angle simply, but this is less simple when you have rings.
But we sample it the same way. That was not any difficulties that we measure. We assign the phi and psi angles the same with the linear and perform and presented this sample in the Ramachandran plot and compare with the available experimental data.
But I mean, if you cannot, the angle are not free of rotating as much as you want. So, and it can affect largely you cannot vary an angle without varying the other one. This was not an issue when sampling.
We actually determined what was the phi angle in our cyclohexane ring and what was the psi angle, and then we performed we calculated these values and produced these Ramachandran plots.
Okay. Thank you very much. I also noticed that in the parameterization, you used the 2 quantum mechanical method. You used both the PVA0 functional and the B3 leap D3.
Yes.
And I was wondering why. And yes.
D3 we use mostly for IB homopeptides because they are more non-polar and because of the presence of 2 methyl groups, the steric hindrance was very high, and we wanted to express dispersion in for this kind of the peptides, and that was the reason. So we performed for the peptides to the B3 with D3 model, and this was some separate work. So first we deal with this amino cyclohexane ring and TOAC deep, and develop force field by using B3. And then when shifted to IB deep peptides, we use more advanced methods for the optimizing the structures of these peptides, since they are very smaller. And with the both of methods, we were satisfied while comparing our results with the experimental available data.
So you wouldn't suggest one or the other, PB0 or PB free lip D3?
Very good. So maybe B3 lip D3 is better.
Okay, so I have other
protein
curiosity.
simulations.
I have other curiosity, but we can speak in other occasions. So I now ask to the audience if they have any question. I see
Yes,
that
yes. We have a question. So, Dr. Pyatnitsky.
Thank you for your lecture. I have a simple question just for my understanding. Am I understand correctly that all force fields are constructed on results of quantum chemistry and validated on experimental data? Am I correct?
Yes, we use a lot of quantum mechanical data and a lot of experimental data. As more as you have available, it is better to compare your results.
So it is quantum chemistry and fitting of experiment, yes?
Yeah. But this is a usual procedure for the fitting parameters.
Thank you.
You're welcome.
Okay, thank you. Are there more questions from the audience?
May I ask, Francesca?
Yes, please.
Sonja, thank you very much for a very interesting lecture, very interesting overview of all these force fields. And I wanted to ask probably a little bit complicated question in sense of force fields. So usually we use the functions, potential functions. For instance, you show, and previously also Francesca shows these potential functions for bonds, for valence angles, dihedral angles, and so on. And we know these potential functions, and we know the force fields which are used, the parameters which are used for these potential functions. But in fact, probably we miss something if we introduce just this potential functions. So I mean, if we take maybe some anharmonic terms, we can expect qualitatively different effects, physical effects. I mean, if we take string with harmonic potential, we can have one behavior, but if the string have some anharmonicity, we can have some unexpected behavior, as it was in the simulation in Ulam pasta single chain. So when anharmonicity induce some unexpected behavior which is not described in a usual way. So probably we should think also about consideration of other potential functions, including some nonlinear terms.
Okay, so thank you, Sergio, for this interesting question. So anharmonic terms, they go beyond Hooke's law, and they allow a more realistic description of molecular vibrations and energy surfaces. especially at large deviations from equilibrium. So what I would say about the force field that I mentioned here, we use mostly harmonic because these are implemented in number, and they are work very well for proteins and amino acids. But if you would like to do some more complicated refinement, then you have to insert new terms in the force field, which is existing one with the new functions, and also to refine the new parameters, which is also possible. But this you have to do mostly manually for when you work with the standard force field. But all these standard force field that I mentioned, they have these harmonic terms.
And there are works in this direction, as I understand. Such approach is used.
So unharmonic, some force fields, especially some polarizable or reactive ones, that they can contain these unharmonic terms.
Okay, thank you very much.
You're welcome.
Thank you, Sergi. Thank you, Sonja. Do we have more questions from the audience? From Zoom? Please raise your hand if you want to ask a question and open your microphone. Okay.